Now we're going to talk about lipids. Lipids are not always fat, but fat is always a lipid. We have other things in the human body that is considered a lipid that are not fats, but they are very important to our life. We have hormones, we have phospholipids, we have many other structures, possibly things I don't even know about. You have to have some lipids in your body. You can't be completely lipid free. That's not a thing. You have lipids that circle your myelin sheaths. Well, no, I'm sorry. They're called myelin sheaths, and they encircle the neurons of your brain to insulate it and help with electronic nerve propagation. So if you want to have proper neuron function, you have to have some lipids. You are supposed to have, at the very least, a small layer of lipid under your skin for protection and insulation. You have lipids, as I said, in every single cell membrane of your body, and you are made up of many cells. You also have lipids, once again, as hormones and chemical signalers. So to have fat in your body is not wrong. To have too much fat, then you start playing games with cardiovascular disease and hurting your knees because it's carrying too much weight and not being able to properly utilize your body. But as I say, all fats are lipids, not all lipids are fats. So let's get into lipids, fats and oils as we know them. Hey, I need to share my screen. Ha-ha, let's go back. Here we are. Lipids, share. There we go. And once again, I'm going to minimize myself. Okay, so lipids, as we know them, fats and oils. You have many infused olive oils in this picture. Here is butter in many formats. I'm assuming this is real churned butter from straight cow's milk and not processed butter. Peanut butter, almonds, cashews, they all have natural fats in them. You have natural fats in things like beans and lentils that are actually quite healthy for you. And then you have real mayo, which is an emulsion of egg, and I'm not even sure what, egg and oil, I'm pretty sure, that makes a spread. Not typically the healthiest thing on the menu, but also probably not the worst thing on the menu. Lipids. I don't know when you all were born, 2003, 2004? That sounds insane to me, by the way. There used to be a TV show, it was a cartoon, actually, called Fat Albert. And his catchphrase was, hey, 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 you know, so whatever. And then here's all the butters in the world. And we do many correlation studies when we are trying to figure out human health and how humans utilize things. We typically start with mice. And so here you can see we were doing, not we, I wasn't, but somebody was doing an obesity study on mice. And look how large this mouse is compared to a normal healthy fat mouse. Lipids are long-term energy storage. Recall, please, that carbohydrates are quick energy storage. Lipids are long-term. We store them away, planning to use them in case. They are our in-case storage. If we need them, this is where we can find the energy. And it's concentrated energy. We'll find out in a couple of slides that it's actually 2 times the amount of energy as carbs, but it's harder to get to. Just like carbs, lipids are made up of carbon, hydrogen, and oxygen. And that's basically where the similarity ends. They form long hydrocarbon chains. So a majority of what lipids are, remember please that carbohydrates had a whole bunch of oxygens in them. Almost every single carbon has a hydroxyl group hanging off of it with an oxygen. Lipids very much concentrated on the hydrocarbon part of it. Excuse me, so sorry. Very much focused on the hydrocarbon part of it. So lots of hydrogen and carbon, and very little oxygen. They have family groups. You have your fats as we know them, fats as we eat them. This is a triglyceride. It is your common fat. It's 3 hydrocarbon chains connected to a glycerol backbone. And you can see the sheer amount of carbons and hydrogens versus the oxygen that is in a triglyceride or a fat. We have phospholipids. These are important for membranes. We have steroids. These are our hormones in our human body. You have steroids like cholesterol, which is going to be the only one I can remember, and it's the most important one that we talk about in terms of this class. Cholesterol, funnily enough, is also embedded in phospholipid membranes as well. Not like carbs, proteins, or nucleic acids, lipids do not form polymers. We do not get long chains of lipids. We get large molecules of lipids that are made up of smaller subunits. Take this triglyceride, for example. You have 3 fatty acid subunits, and you have a glycerol backbone subunit. Put it all together, you get a large molecule of a triglyceride. It is not a continuing train. We're not going to add on another triglyceride to the end of this and then add on another triglyceride to the end of that. This is the entire structure. Now, having said that, this hydrocarbon chain can be much, much longer than as depicted here. And that would be a different type of fat. But we don't add them and add them and add them together like we do with carbs and proteins and nucleic acids. So let's talk about fats. As I've said, it's a glycerol backbone. It's 3 alcohols, 1, 2, 3. On carbon 1, 2, 3, you have alcohols hanging off of it, or hydroxyl groups, and a fatty acid chain. The fatty acid is a long hydrocarbon tail with carboxyl group at the head. So here's our 2 functional groups in a fat, a triglyceride specifically. You have a hydroxyl group, and it's going to do a dehydration synthesis, and you're going to lose water and bond to what was a carboxyl group. Remember, a carboxyl group has a carbonyl, and it's at the end, so that would be an aldehyde. I don't know why I winked at you, but that's what it would be. And then it also has a hydroxyl. So you're going to lose this hydrogen, and you're going to lose this hydroxyl as water, and you're going to get COC double bonded O. Lose water. Of course, it uses enzymes. And remember, I told you every single bond that we form through dehydration synthesis, if it's a carb, lipid, protein, or amino protein or nucleic acid, is going to have a different name to that bond. Carbohydrates are glycosidic linkages. Lipids are ester linkages. But see here, it's that carbon, there's that oxygen that was a hydroxyl group. Here is this carbon that had a hydroxyl group coming off of it. We lost a hydrogen from here and the hydroxyl group from here as water. It's gone, dehydration. And you form this ester linkage. And this is a triglycerol. We typically call them triglycerides. 3 fatty acids linked to a glycerol backbone. Take a minute. Excellent. In case you needed to remember that that's a hydroxyl and that's a carboxyl. Here is the same thing, just showing it all together. Lose that hydrogen, lose this hydroxyl, there goes water, dehydration synthesis, ester linkage, ester linkage, ester linkage. All through the use of enzymes. Fats store energy, this long hydrocarbon chain, those bonds between all those carbons, that's energy. Are fats polar or non-polar? Well, that's exactly right. They are non-polar. Not for nothing. I mean, they do have these two oxygens in here, which would make you think maybe they interact with water. But this long hydrocarbon chain has no interest in interacting with water. They have no charges among them. The whole structure ends up being nonpolar. If you are a nonpolar molecule, are you hydrophilic or hydrophobic? Astounding. Yes, it's hydrophobic. Very good. The function of these nonpolar, hydrophobic, long fatty acid chains is energy storage. It's concentrated. It's all hydrocarbons. It's 2 times the amount of carbohydrates. But it's not quick. It takes a lot to break them down. It cushions our organs, and it insulates our body, and it's used in our neurons. It's everywhere. Think of whale blubber. I was going to try to make a whale noise, but then I got embarrassed. So, I don't know. That's more like a cow. Why do humans like fatty foods? It's an evolutionary trait. Just like we tend towards carbohydrates as our favorite thing to eat because it's quick energy, we like fatty foods because evolutionarily speaking, we quite didn't kind of know where our next meal was coming from. And so it was good to have stored energy. Now you have to think of the body of a Homo sapien, Neanderthals were not Homo sapiens, but of a Neanderthal man. As they are depicted currently, there was not excess fat on those humanoids because they burned it all. They had an energetic lifestyle that burned the fat that they consumed, but they were willing and happily ate fat because it's long-term energy storage. We have 2 types of fats now. I mean, they've always kind of existed. But we have something called saturated fats, and then we have non-saturated fats. In terms of health, in terms of consuming, saturated fats are going to be the worst thing you can consume if you overdo it. Saturated fats do have a role to play in our diet. I don't know where I was going with dietary. In our diet, there are supposed to be some saturated fats in your diet, just not at the level that we typically consume them in, let's say, America and Europe and some South American countries. Saturated fats are all carbons bonded to all hydrogens. That means every single carbon is forming 4 separate bonds, 2 to hydrogens and 2 to the other carbons they're attached to. There are no double bonds in saturated fats. What this means, so I'm specifically talking about fats like this. If you look at the carbons, carbon 1, 2, 3, 4, carbon 1, 2, 3, 4, carbon 1, 2, 3, 4. Every single carbon is sharing all 4 electrons with 4 different other atoms. So they're all single bonds, and every carbon is completely loaded with as many hydrogens as it can carry. That is a saturated fat. These end up being long, straight chains, straight being very important. And because they're straight, they can pack together on top of each other very, very tightly and form blocks of fat. And these are most of our animal fats. You see animal fat on meat products when they're raw. I mean, you see it when they're cooked too, but it's easier to identify when they're raw. If you're taking chicken out of a package, it has white fat hanging off of it. You can see it in marbled steaks, for those of you who love to eat your steaks. You have steaks that are very lean. I'm not very well versed on steaks, but like a sirloin is lean compared to, I had it in my head for a second. But there's other steaks that are highly marbled. That marbling, which means it has white streaks running through the raw meat, is actually all animal fat. It makes the steak more tender, I believe. They're solid at room temperature. Eating too much saturated fats leads to cardiovascular disease. Mostly through atherosclerosis. Athero means hardening due to plaque deposits. Now, Sorry, that was wrong. Here I am, a medical terminology teacher. Athero means fat. Sclerosis means hardening. So what happens is the fat lines the blood vessels, and so the blood vessels then can't function like they normally can, beating with the pulse, because that fat is restricting their movement. So your solid butters, your margarine, your, well, you think I'd know more fats. I mean, have you seen me? Actually, y'all haven't, have you? Not this year. Anything that's solid at room temperature is a fat. And then you have your unsaturated fats. Unsaturated fats means that not every single hydrogen, no. Going back to this picture. It would mean that this carbon, let's say our fourth carbon, is missing this hydrogen down here at the bottom. So it has to form a double bond to get all 4 of its electrons that it needs. And then down here, this carbon could be missing this hydrogen. So it has to form a double bond, okay? So unsaturated means it doesn't have all the hydrogens that it can hold. These carbon-to-carbon double bonds cause kinks. They're called kinks in the fatty acid. These kinks cause the fatty acid tails to be pushed apart, which means they can't pack very tightly like saturated fats can. Plant and fish oils or fats, vegetable oils, these are liquid at room temperature. The kinks made by the double bonds the double bonded carbons prevent the molecules from packing tightly together. That's not all. Then you have monounsaturated versus polyunsaturated. Monounsaturated being only one double bond occurs in that entire fatty acid. Polyunsaturated meaning many double bonds occur. In terms of health, you want to eat polyunsaturated fats. Not only do they not pack so tightly in the human body, but it takes much more energy to break a double bond. And so if you need to utilize the fats, you are using energy to break down the fats. And so you won't store as much fats. So here is a saturated hydrocarbon chain. Here is a monosaturated hydrocarbon chain because it only has one kink in it. And in terms of health, you don't want to eat saturated. You want to eat unsaturated. Okay, so those are fats. We have phospholipids. Phospholipids are essential to our life. You can have all sorts of different types of phospholipids, but they all have to do with cell membranes. The structure is a glycerol. Oh, sorry, that's down here. A glycerol with your carbon chain with hydroxyl groups coming off of it, and then 2 fatty acids. And one of them was almost always unsaturated. So then you have 2 fatty acids and a phosphate group. Remember, in terms of functional groups, phosphates hold a lot of energy. It's just like a penguin, a head at one end and a tail at the other. Except penguins have very small tails, so I would say that these are more the penguin legs. So if you look at it, it's either a really creepy clown face, here's the eyes and here's the nose, and then they don't have arms and they only have legs. That clown sounds terrifying. Or it's a penguin with red pieces. Phospholipids, hey, are phospholipids hydrophobic or hydrophilic? They are both. They're both. Hydrophilic head right here, hydrophobic tails. This is something we call amphipathic. Let me write that down and share it with the screen. Amphipathic. A-M-P-H-I-P-A-pathic. Now I just had to confirm that I was spelling it right. I don't want to give you a wrong spelling. But amphipathic, A-M-P-H-I-P-A-T-H-I-C, is to have both hydrophilic and hydrophobic parts, like phospholipids do. When a phospholipid is forming a bilayer, what happens is you have a phospholipid head and another one and another one. We'd show the heads just by circles, right? And then you have the tails, and you usually do that. Okay. But a phospholipid bilayer implies two sides to it. So that's actually doubled up, with the hydrophobic heads always facing the outside and the hydrophilic tails all inside. And so this is facing the extracellular environment, and this is facing the intracellular environment. And this whole region in between is hydrophobic. Evolutionarily speaking, it had to happen this way in order for cells to be able to separate outside from inside environments. So we have this beautiful bilayer phospholipids. So amphipathic is the term that we call phospholipids and other molecules that have the ability to be hydrophilic on one end and hydrophobic on the other. So the phospholipid head is attracted to water, and the phospholipid tails are repelled by water. This interaction with H2O is complex and very important. It has a split personality. So way back in the day, the very first prototypes of cells only had a single lipid bilayer, a single lipid layer, right? Because it's not bi. And that's called a bubble or a mycel. You can see that if you put oil in water and shake it really hard, then all the individual oil droplets that are separated in the water, those are mycels. And it was the very first precursor to cell membranes that separated one side of an aqueous environment from another side of the aqueous environment. Then through evolution and however else you could believe it happened, we ended up forming a phospholipid bilayer. Now, just by the way it's structured with hydrophobic tails, it's probably formed very naturally because the hydrophobic tails are going to look for each other. Like dissolves like is the phrase, right? Fats dissolve fats, and aqueous solutions dissolve in aqueous solutions. So the fats are going to look for each other and not combine with each other because they're not bound together, but they're going to orient towards each other, forming this entire hydrophobic layer in between an extracellular aqueous environment and an intracellular aqueous environment. Yeah, and mycels were most likely the earliest evolutionary stage of a cell. Also, well, it doesn't matter right now, but the first type of DNA inside early cells was RNA. That's a 1407 thing. Why is this important? Why do we care that phospholipids face outside and have an inside hydrophobic layer? It creates a barrier, which is extremely important in order to perform life necessary functions inside a cell without it being disrupted with whatever's going on extracellularly. It's important to be able to intake nutrients but not harmful substances and remove waste without losing all your nutrients. They make cell membranes. See here, see here, see here. There it is. Soap. How does soap function? It does wash your hands. What else does soap do? It does bubble up. What do you think that bubbling up means? What soap actually does is it disrupts the oils or other solutions that are on the surface of your hands. Soap doesn't do anything to the bacteria or viruses or fungi or whatever on your hand, but it disrupts the oil layers that could be holding them in place, which allows water to then wash them off your hands. Soap makes water wetter. Okay, think about that for a minute. Soap allows water to come in a stronger contact with your skin to wash off bacteria, salts, things from your sweat, things from your oil glands, to actually get them off your skin. The soap is not doing it. It's just allowing water to do it more efficiently. Get all this up here. Okay. So the phospholipids of a cell membrane, of every single cell membrane that exists in your human body, is a double layer or a bilayer. The hydrophilic heads are on the outside in contact with the aqueous environment, monitoring what's happening both inside and outside the cell, letting some things through, not letting other things through. The hydrophobic cells are on the inside, forming that barrier so that you can have different processes occurring outside and inside. You have your hydrophilic region, you had your hydrophobic region, and you have another hydrophilic region. So let's look at this picture very closely. If this is hydrophilic, what about these proteins that are sticking out of this area? What does this region of the protein have to be? It also has to be hydrophilic. But then if you cut through a cell membrane, you see that these proteins are embedded completely through that cell membrane. So what does this region of the protein have to be? Right, this has to be hydrophobic, which means these proteins have to specifically form to have a hydrophilic region on the inside, a completely hydrophobic core, and then a hydrophilic region on the part that's sticking up on the outside as well, right? The third class is steroids. The structure is 4 fused carbon rings plus whatever's hanging off of them. Different steroids created by attaching different functional groups to the rings. Remember, the functional groups change the physical properties of what that's going to be, what that hormone steroid is going to be able to do. The different structure creates a different function. Examples are cholesterol, which is this, and our sex hormones, like we talked about in the carbon lecture, how testosterone and estrogen are actually so similar and just very minutely different. So here's cholesterol. You're supposed to have cholesterol, not a whole lot of it, but you are supposed to have some of it. It's actually part of your phospholipid membrane. Too much leads to atherosclerosis, as seen here, those fatty plaque deposits that harden your arteries. But cholesterol is important in cell membrane, and it's the precursor of all other steroids. You just can't have too much of it. Your blood work shows you that if you, I don't know what age they start taking blood yearly, but at some age they start taking blood at your yearly physical and then tell you, hey, your cholesterol is too high or too low or just right or whatever, and they might put you on cholesterol medicine. So it's an important component of cell membrane. Whoops. It helps keep cell membranes fluid and flexible. It breaks up the phospholipids. It's embedded into the phospholipids. You see here all these methyl groups, right? All these CH2 groups are going to be hydrophilic or hydrophobic? Hydrophobic. But you see all the way down here. So this is CH, 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 another methyl group, but here is a hydroxyl. Is that hydroxyl going to be hydrophobic or hydrophilic? It's hydrophilic, right? That oxygen has a partial negative charge, and so it's going to willingly interact with water. This is another amphipathic structure. So it's embedded in the bilayer, the membrane, and it breaks up all the phospholipids, allowing it to be more fluid. They actually sometimes form things called lipid rafts. Now, there's a caveat to this, that you can actually have too much cholesterol in a lipid bilayer, in a phospholipid bilayer, and too much actually makes it more rigid. Because one thing that you don't know yet is that these phospholipids are constantly moving. They're flipping against each other like this constantly, and they're moving around the cell while changing places with the ones near them. They can actually move around the entire cell in some form of seconds that's actually too small to actually like be comprehensible. And if you have too much cholesterol stopping that movement, then the fluidity of the cell membrane slows down. And then, as we said, sex hormones. So here is oh, these are steroids. These are sex hormones down here. That makes a lot more sense. These are components of these are steroids made in your adrenal glands that are on top of your kidneys. Double bonded O here, a carbonyl, here's an OH group, and then just slightly different. You have nearly the same structure here. Instead of having a hydroxyl group coming off of this carbon, you move down to this carbon and have a carbonyl group coming off of it. One of them is a glucocorticoid and another one is a mineralocorticoid, and they have different functions in the human body. And then once again, testosterone and estrogen or estradiol, which is the precursor to estrogen. These are methyl groups. See, they don't put the carbons here. They don't put CH3, CH3. They just are indicating that there is a carbon hanging off of there. So you have the same hydroxyl and methyl group right here, but then the testosterone has an extra methyl group and a carbonyl, while the estrogen has a hydroxyl group. And then progesterone is typically female-only hormone for birth, reproductive purposes, and it's almost the same structure again. It's actually very near testosterone, but then you change this structure. So look how close all of our hormones are, so close related, and just the functional groups change what they do. Oh, hit a circle level. All right, there we go. So there you go. Fats, phospholipids, and steroids are the three classes of lipids that we have in the human body that are important to our life functions. Gonna keep smiling. Goodbye.